Alright, so we've already covered binary fission, and now the other type of asexual reproduction is mitosis. But before we go into mitosis, it is essential that you know what the basic cell cycle is. So, I didn't even show you my card. The cell cycle. Alright, so the cell cycle essentially looks like this. If you look at it, we've got 4 chunks to the pie, right? So we're going to start right here at this line, and that's going to start one cell. I know this is like the worst schematic ever, but here you go. So we'll start here. We have one cell, and before it can even go to mitosis, which is M right here, it has to go through G1, S, G2 phases. All right? So we're going to go through, and I'll briefly explain what each of these little phases are. And you can notice that each of them have like a set amount of time dedicated to each one. Even mitosis is a little bit large. Mitosis is like the smallest little sliver of the pie relative to all of this preparation. All of this, G1, S, and G2, are considered parts of interphase. Interphase is not part of mitosis. That might be a question on your exam. Is interphase part of mitosis? The answer is no. It's part of the cell cycle, yes, but is not part of mitosis, okay? So those are your phases. I'll bring this picture up every so often. Alright, so the first phase, before we can do any mitotic division, is G1, the gap phase. And I put forever because it literally lasts forever. There really isn't a set time period when G1 phase will actually end because it just goes on forever. And in fact, I didn't write a card for it, but I wrote it on here. You can see it says G0 off to the side there. There are some cells, like liver cells, that actually go into a phase called G0 phase, where they actually indefinitely remain in G0 phase. And the cool part about G0 phase is that it's actually, we can induce G0 phase in some cells, meaning they won't ever divide. And that's kind of like where we're going with cancer medication right now. It's like trying to put it into G0 phase so they don't divide. Anyway, so cells in G0 phase over here never ever go on to S phase. They can be pulled out of it by a signal, but other than that, they are totally stagnated. G1 is a gap phase, meaning it just kind of like sits there, it's part of interphase. It kind of gets everything in line. Bob, my biology teacher in high school, he called it putting your ducks in a row. Get your ducks in a row before you can go on to synthesizing. This is the longest phase because if anything is wrong with the genetic machinery, with any of the enzymes that are used in synthesis, if anything's wrong with that, the synthesis of the DNA, S phase, everything's just going to fall apart. Because if you screw up synthesis, your cell will die if you have any big chunk of that missing. Does that make sense? I don't know why I'm asking you because you can't respond, but alright, so G1 is the longest because it has to get everything right, like perfectly. And if it doesn't, it just kind of stops and we'll start over again so we don't fuck up. Okay? After G1, we have a pie slice. So right here, now we're here. We literally reach a checkpoint. The checkpoint is super important. This is like what I was saying. If anything is wrong or out of place or missing or mutated, the cell will know most of the time. There are a few instances where it won't notice and then the cell will die. But if the cell realizes at this checkpoint that, oh crap, we don't have enough of this resource, it will start over and get its shit in line. Okay? Does that make sense? Alright. After it gets its duck in the row and it approves the checkpoint, it moves on to the S phase. See, S is the next phase right here. S stands for synthesis. That's it. We synthesize the DNA, and that's all that happens. But it takes a long time. You have to think about how many base pairs are in DNA. I mean, it varies from creature to creature, but there is a lot of DNA to go through, and it takes a lot of time. So we wait for synthesis to be over, and mind you, this is still a part of interphase. All of this, from this end slice to this end slice, all of this is interphase. Okay? So synthesis phase, the S phase, is also part of interphase. Alright, so that's just synthesizing DNA. The next one is another checkpoint. You can see here, my little schematic here, that I wrote another checkpoint right underneath G2, my beautifully written pie chart. It's the same reason. If for some reason our machinery just totally screwed up the synthesizing process, we don't want the progeny of that cell. We don't want it, because we know it's mutated, we know it's wrong, we know there's chunks of it missing, something's bad. We don't want to create a cell that is not viable. So it will terminate the cell or terminate the process depending on how far along things are. So there's a checkpoint between like each side of the S phase, before and after the S phase checkpoints. Super, super important. Okay. After the S phase and the checkpoint, we move on to gap 2 phase. And essentially what gap 2 is a lot shorter than gap 1 because we've already done most of the most complicated stuff. Gap 2 is essentially just getting everything ready for mitosis. Gap 2 is still a part of metaphase, and basically getting ready for mitosis means like preparing all the microtubules that are needed for the rest of mitosis. Anything that is needed to carry out the process of mitosis is being prepped for or during gap phase. Okay, so gap 1, we prep everything for synthesis. Synthesis is actually synthesizing DNA, and gap 2 is prepping for mitosis. Okay, and then finally, we've done G1, S, and G2. Now we're finally to mitosis, which I put little letters on top of there too. And I'll give you my acronym later, to like your little memory trick later about mitosis. But then we finally reach mitosis, M phase, which is the actual dividing of all of the genetic material, all of the machinery, all of the enzymatic components of the cell into 2 individual daughter cells. So that is the basic cell cycle.